having me here. So in the next 10 to 15 minutes, I'm going to give you a very brief summary about a statistical approach for designing and conducting real subtype clinical trials. So real subtypes often refer to lung clear cell RCC, which only accounts to about 20 to 25% all kidney cancer cases. But we know this is not a single disease. It includes multiple histological subtypes, each with unique bioenergy and clinical characteristics. Because this diversity and rarity, it's very challenging to conduct a prospective clinical trial in all phases of the drug development. The phase 1 study struggled without robust biomarker to guide a subtype-specific drug development. For phase 2 study, we need to address heterogeneity despite very small sample sizes in the phase 2 setting. Phase 3 trial is often not feasible because slow patient accrual for many real subtypes. Also, it's difficult to define a control arm in the absence of established standard care for many subtypes. A phase 3B or phase 4 study could also test drug on lung clear cell RCC after drug has been approved or on the market. This type of study often use a descriptive design and primarily enroll community-based patients. The sample size was often based on feasibility. For lung clear cell RCC trial, patient selection is critical. There are 3 different approaches, each with its own advantages and limitations. The first approach is all-inclusive approach or mixed bag approach, which includes all lung clear cell RCC subtypes. This approach offers 2 key benefits. Fast patient accrual also results with a broader application. However, the trial will enroll very heterogeneous population. Also, if different subtypes respond to treatment differently, the overall treatment benefit might be diluted. This approach has been used for immunotherapy-based trials or early trials which focus on safety or preliminary efficacy. The second approach is a subtype-specific approach, which focuses on individual variants. This offers more homogeneous population. Also, results has a cleaner efficacy signal. However, accrual could be challenging for many real subtype lung clear cell RCC. Also, results can only apply to particular variants and cannot translate to all lung clear cell subtypes. This approach has been used for targeted therapy-based trial. The third approach is a biomarker enrichment approach, which targets a bioenergy-driven responder. If the right drug is matched with the right bioenergy pathway, it will increase the likelihood of observing clinical benefits. This approach also aligns with personalized medicine. However, it requires comprehensive pre-screening of biomarker to identify eligible patients. Also, it demands complex logistics and infrastructure for the trial operation. This approach has been used for MET mutant and targeted therapy-based trial. For lung clear cell RCC trial, we also need to choose appropriate design. The single-arm, one-stage design offers simplicity. It requires small sample size and enables fast completion of the study. But there is no early stopping rule if the drug has limited activity. The single-arm, two-stage design adds early futility analysis to minimize the number of patients exposed to inactive drugs. However, if the trial includes multiple histological subtypes, there is risk of premature termination if non-responding infavorable subtypes are overrepresentative in the early stage. The multi-arm randomized design provides the most robust comparison between treatment. It also allows histological subgroup analysis. However, this requires very large sample size, which may not be feasible for many real subtypes. In terms of drug development strategy, the lung clear cell RCC trial generally follows the approach used in clear cell RCC. For many years, the targeted therapy, particularly VEGF-TKI and the MET inhibitors, has been the treatment backbone for lung clear cell RCC. This has been shifted recently. For now, the current focus is immune checkpoint inhibitors and their combination with targeted therapy. Here is a review of the published lung clear cell RCC trial in the last 10 years, which includes 22 targeted therapy-based trials and 13 immunotherapy-based trials, either ICI alone or ICI in combination with targeted therapy. Regarding trial characteristics, for both targeted therapy-based trial and the ICI-based trial, the majority of the trial use the phase 2 single-arm design. And the median size is about 40 within the single-arm phase 2 range. About patient characteristics, most of the trial require low prior therapy or only up to 1 line of prior systemic therapy. Regarding histology, for targeted therapy-based trials, almost half of the trial focus on papillary RCC. But for ICI-based trial, majority, 85%, including multiple histological subtypes. Regarding number of patients by histological subtypes, for targeted therapy-based trial, nearly 80% were papillary RCC. For other lung clear cell subtypes, the number is generally low. For ICI-based trial, we say about 60% were papillary RCC, chromophobia 17%, unclassified 11%. Here, I also want to highlight a few ongoing randomized phase 2 or phase 3 study. The Steiner 304 is a phase 3 study which will include multiple histological subtypes. The other 3 studies, Sumanta, PEP-MET2, and PEXMAM, only focus on papillary RCC. You will see all these randomized trials evaluate the combination of target therapy with immunotherapy. That's the current focus. There are also ongoing basket of trials which may involve lung clear cell RCC. Basket of trial test a drug across multiple tumor types which share a common genetic alteration. For example, the NCI match trial is a basket of trial which evaluates multiple target therapy based on specific genetic alteration. The MET mutant papillary RCC could be eligible for one of the matched arms. The SWOG 1609 is another basketball trial which assesses the combination NIVA plus EP across multiple real tumor types. One of the cohorts focuses on spindle cell carcinoma of real GU tumors. There is also ongoing trial, phase 2 study, which utilize a Bayesian optimum design. This is a phase 2 single-arm phase 2 study, 14, which is a phase 2 study to evaluate the efficacy and the safety of the combination therapy nivolumab plus nivolumab in patients with advanced lung clear cell RCC who had received up to 1 line of prior systemic therapy. Regarding study design, the study primary outcome is objective response rate. The total accrual is 48 patients. Patients will be enrolled in 4 stages with interim analysis for efficacy and safety after every 12 patients. So this design offers flexibility in terms of the interim analysis timing and could reduce sample size potentially. In summary, so the current lung clear cell RCC trial largely mirror approach used in clear cell RCC, and most study conducted in the phase 2 setting. And the primary focus were papillary RCC, especially for the targeted therapy-based trials. In the future, to address the unmet need of this real and diverse disease population, we need a robust biomarker to guide the subtype-specific drug development. We also need a more innovative design to improve the efficiency of clinical research. We also need to build collaborative research networks to optimize resource use for this real and diverse disease population. Thank you for your time.
Okay, we are going to continue with the live speakers, and then I think we've corrected the audio issues, but we'll do that at the end. So we're going to continue with Joshua Mann, who's part of the VHL Alliance. Thank you so much for being here, Joshua.
Good morning, everybody. My name is Joshua Mann. I'm the Director of Health for the VHL Alliance. Thank you so much to the organizers for having me, as well as everybody who came from out of town to visit us here in Boston. Although I've lived in Boston for about 10 years, I'm still a New Yorker at heart, so you won't hear any jokes from me like Pak the Ka and Harvard Yad or anything like that. And I'll be talking about patient advocacy, registry, and trial recruitment, what has worked in rare cancers, especially for the VHL Alliance. And there's my email address on the bottom if anybody's interested in reaching out. So a little bit about the VHL Alliance. The VHL Alliance was founded in 1993 by patients and families affected by VHL. Our mission is to improve the quality of life and health outcomes for VHL patients, families, and caregivers through inclusive community building, connections to excellent education and treatment options, and advancements in medical research. Our vision is to cure cancer through VHL research. No simple task. We've grown to become the leading patient advocacy organization for VHL, and we play a key role in advancing VHL research and clinical care. We also advocate for rare disease funding and policies that benefit our community. This has resulted in over $7 million in CDMRP grants awarded to VHL researchers for their research this year alone. Despite being such a small organization and a small community, we are a significant force multiplier in the cancer and rare disease space, particularly concerning VHL. In fact, 9 different approved cancer drugs have come out of VHL research, the most recent of which being belzutifan, which I'm sure everybody here knows a lot about. So a little bit about VHL. VHL's disease is a rare inherited disorder. It causes tumors and cysts to grow in various parts of the body, and the incidence is about 1 in 36,000. We believe there to be about 10,000 patients in the US with VHL disease. It's caused by a mutation in the VHL gene, which is a tumor suppressor gene, and it follows an autosomal dominant inheritance pattern. So it affects families significantly. The tumors can be benign or malignant and can develop in up to 10 different organs, including the brain, spine, eyes, kidneys, pancreas, and adrenals. Early detection and regular screening are crucial for managing VHL and preventing more serious complications. Early on, the VHL Alliance recognized a critical need for comprehensive patient data in VHL. So to address this, about 10 years ago, we launched the MyVHL Patient Natural History Study. We were the first patient advocacy organization to launch a registry on NORD's I Am Rare platform. It's a longitudinal patient-consented online natural history study that was originally called the Cancer in Our Genes Project. Our aim with MyVHL is to collect long-term, real-world data on VHL progression. The data points we collect include demographics, genetic mutations, clinical manifestations, treatment histories, surgical interventions, and outcomes. We also collect quality of life measures directly from the patients themselves. Currently, MyVHL has 1,350 total registrants with 1,094 consented participants who have collectively submitted and are responsible for over 13,000 survey submissions. So what about clinical trials? Now, there are several barriers to consider when recruiting for clinical trials in rare disease and rare cancers. These include small patient populations and geographic dispersion of patients globally. So in the case of VHL, patients are spread all around the world and all around the country, with no individual group affected more than another. There's also a general lack of awareness about trials among both patients and clinicians, and strict inclusion and exclusion criteria can limit participation. Patient advocacy groups like the VHL Alliance play a vital role in overcoming these barriers and instilling trust and confidence in patients and families. We conduct direct outreach to our patient communities, serve as a trusted source of information and support, and facilitate crucial connections between patients and trial sites. Centralized registries such as MyVHL are also incredibly valuable. MyVHL allows us to identify eligible patients based on study criteria and enables streamlined communication with interested participants, which in turn reduces the burden on individual sites for patient identification and speeds up trials. We also have the capability with MyVHL to quickly roll out new surveys targeted to subsections of the community as needed. So, best practices. Our approach to recruitment is patient-centric. We ensure that trial information is clear and accessible to everyone. We emphasize the benefits to patients and highlight the invaluable contributions they make to research. We also strive to minimize the patient burden by offering remote monitoring and travel support whenever possible, or connecting patients to those kind of support resources. So some key takeaways. Patient advocacy, led by organizations like the VHL Alliance, is absolutely crucial for cancer research, especially with regard to rare disease. Patient registries, such as MyVHL, are invaluable resources for collecting natural history data and for efficient patient recruitment. Ultimately, collaborative efforts between patients, advocates, clinicians, and researchers are what truly drives progress in rare disease and rare cancer. Thank you so much for your time and attention, and if anybody's interested, I'd be happy to answer any questions.
Thank you so much.
Thank you very much, Joshua. And I think his points were very well taken, and I congratulate Srini for doing this in translocation, these types of concepts, but I think this is something that we should all be thinking about for rare histologies.
All right, our next speaker is Dr. Monty Powell from City of Hope, discussing the role of industry in conducting rare tumor histology trials. Thank you, Monty.
Great. Thanks so much, Rana. I really felt bad for myself because I got in at 2:30 in the morning, and then I found out that Rana actually got here at 5:15 from the West Coast. So in any case, it's an interesting topic, industry support of rare subtype trials, but I do think it's an important conversation and certainly a terrific conversation to have in this room full of experts and likewise folks who are entering into the field. I just wanted everybody to do a little bit of a visioning exercise with me for a second. So I've been at City of Hope for about 20 years now, and I've been seeing patients with kidney cancer for just as long. This is a diverse audience, so I think we've had individuals in the room here who have been seeing patients for longer than that, some who are relatively new to the field. But if you think back to maybe 12 to 15 years ago, you might remember this landscape where we were starting to see, in a treatment horizon where we principally use drugs like sunitinib and pazopanib, some of these single-arm studies, right? And we had these beautiful waterfall plots that were coming out. I think at that point in time, there might have been some temptation, some penchant, and you'll see where I'm going with this, to just take these regimens and just utilize them right up front in clinical practice for our patients with clear cell renal cell carcinoma, right? But I think we did the right thing. If you look at the origin of these waterfall plots, you see the first is related to axitinib and pembrolizumab, the second axitinib and avelumab, the third for lenvatinib and everolimus. And we really utilized phase 3 clinical trials in order to contextualize this data and come up with the very robust treatment algorithms that we're discussing today in this room. So what are the arguments against doing big randomized trials in the context of non-clear cell renal cell carcinoma? I'll walk you through what I think some of the arguments are and perhaps what I envision as being some of the fallacies here. So the first is that we already have approvals in clear cell renal cell carcinoma. These are broad labels. We can already at this moment actually take these regimens and just throw them into patients with non-clear cell disease. I don't really need to spend much time making this argument. I think we heard from the terrific first session today that covered papillary, chromophobe, other histologic subtypes, that non-clear cell subtypes have a very distinct biology. And in fact, I think Wanling and I used this very same diagram to outline that. So I'm going to focus my attention on the second argument here, which is that single-arm phase 2 trial data is very compelling indeed sometimes. You saw that in the first set of slides that I showed you for clear cell renal cell carcinoma. But I want to think back to how many times we've been burned by carefully selected single-arm studies. I put up the data from the pivotal studies in clear cell RCC here for some context, right? And you see what phase 3 randomized trials have done for us, right? You look at the regimens of axitinib and avelumab, you look at bev and atezolizumab, and doing the phase 3 studies, doing things the right way has really allowed us to put this clinical data, I think, into very appropriate context. You know, we can actually lean on perhaps some of these single-arm experiences in other disease types. So, you know, I'll use lenvatinib and pembrolizumab, bev-atezo, and other histologies as one example. If we look to the LEAP series of studies looking at lenvatinib and pembrolizumab, you can see examples in the context of ovarian cancer, endometrial cancer, others where, you know, the data's really been sort of mixed, right? So we really can't take a singular regimen and throw it across multiple histologies willy-nilly. I really do think we need to lean on big randomized trials to establish clinical practice. Now, I will say that, you know, this is a phenomenon that's actually been well studied. You know, as I was preparing for this talk today, I came across multiple explorations of situations in which phase 2 clinical trials don't necessarily translate into positive phase 3 results. And there's a huge host of explanations for that. But, you know, a really, I think, compelling argument comes from the FDA to really push for randomized studies, emphasizing the fact that when we do phase 3 clinical trials and when we really vet the efficacy of regimens, it really lends itself to the fact that patients outside of clinical trials don't get subjected to drugs that would not benefit them or have unnecessary serious toxicities, right? I mean, financial toxicities is another thing to consider. We really have to understand, for instance, in the context of non-clear cell subtypes, what that IO component is really offering here. There really is a pressing need, I think, for us to do these randomized studies. The last argument that I'll take on here, given the short amount of time, is that enrollment is too challenging in non-clear cell renal cell carcinoma studies. But I make the argument here today, perhaps for the first time, you know, that we really can do phase 3 trials in non-clear cell kidney cancer. Tony and Tom and others have, you know, finished the Sumanta trial, which I think is an incredibly important experience, and shown with what I think is a relatively challenging design to implement, that these studies actually can be done. But of course, you know, the question that I've been asked to address today is whether or not we need support from our colleagues in industry. And I think the answer is unequivocally yes. This is a slide that I've shown in previous talks to really sort of summarize the current landscape of clinical trials in non-clear cell kidney cancer, randomized studies specifically, and Wanling in her very elegant talk outlined these studies, PAPMET2, PAXI-PEM, Stellar 304, and Sumanta. And just at first glance, right, if you take a step back and look at this slide, one of the things that you can really identify is that the trials up top are smaller randomized phase 2 studies, and if you look at the point of origin of these trials, right, these are ones that we're trying to take on either as ISTs or studies within the cooperative groups, right? That's one common thread between them. On the other hand, the phase 3 clinical trials that we're attempting are really supported by our colleagues in industry, right? We really do need that gravitas to get behind those studies to get them done. So what if we just try to go it on our own? You know, I wanted to maybe do a little bit of a mathematical exercise here. This is about as advanced as my knowledge of math goes. I can do simple division here. So, you know, I first pulled this very important paper published by Dr. Linehan, Dr. Srinivasan early in the year. So this looks at bevacizumab and erlotinib in the context of both hereditary and sporadic papillary kidney cancer. I pulled from the methods the fact that this trial, which included a total of 83 patients, enrolled from May of 2010 to May of 2019. That's 108 months, right? So if we do the math on this clinical trial, 83 divided by 108 is 0.76 patients per month, right? That's far too slow for us to really sort of achieve answers in the context of randomized studies, okay? So let's move on, and I'll pick on myself here a little bit with the PAPMET clinical trial that Brad had outlined in his talk at the start of the morning, right? So, if we look at this particular trial rather, we started enrollment in April 2016, finished in December of 2019. This was an unselected trial in patients with papillary kidney cancer. And if we do the math on this one, 152 patients over that span of 44 months is 3.45 patients per month. So again, I would argue relatively slow. What's really actually quite impressive to me is that if I look at Sumanta, and I'm just using data that's in public domain here, really not using anything that I've heard from investigators, et cetera, but if I look at that publicly available data, what's really impressive to me is that with this molecularly selected trial, admittedly probably far more challenging to accomplish than a trial like PAPMET, where we're taking all comers with papillary histology, they actually had the same pace of accrual as we did in PAPMET, 148 patients over 43 months. The accrual rate there, 3.44 patients per month, right? So it just tells you that when you put that oomph from industry behind these trials, things can get done a lot faster. And again, just using forward-thinking projections, stuff that's already in press releases or investor calls, we know that Stellar 304 at this point in time is somewhat nearing completion. So if I actually use the projected dates and what I know to be the study start dates here, 291 patients are projected to be enrolled across 24 months. So that's 12.13 patients per month. And this is actually doable. I mean, I think if we have 12 patients going on per month, right, phase 3 trials can happen in the setting of non-clear cell disease. So I think that the numbers here probably serve as the most compelling argument for why we really need industry support. So it's July, and we have a bunch of new fellows, probably some of them are sitting here in this room, who have started in our oncology clinics, and we have all these tenets, right, in medical oncology. The first is that we don't want to extrapolate data from other tumor types, right? We want to produce randomized data that shows clinical benefit before we adopt new standards. I think that's pretty fair. We always recommend they do that. And when there's clinical equipoise, strongly consider enrollment in clinical trials. I'd make the argument that clearly this isn't happening. Today in the rapid oral sessions, my current fellow at City of Hope, Dr. Zugman, is going to be presenting this data through a collaboration with Concert AI. And I think this really articulates the fact that at least here in the US, Concert AI is a database that includes millions of US-based patients. We're really getting ahead of ourselves a little bit. So if you look at this graph here, you can see that the rate of utilization of tyrosine kinase inhibitors has fallen dramatically over the course of time. You see some modest uptake of use of MET inhibitors, but there's this huge rise in TKI-IO combinations in this context. So this suggests that we, at least here in the States, are using that laxity that we have in labels for non-clear cell histology, but maybe not necessarily with cause in all cases, right? Because again, I would argue that we don't have randomized data. What do people often lean on? Well, they often lean on real-world experiences. This is something that I and, of course, dozens of other sites here in the room contribute to. This is the IMDC assessment of outcomes with VEGF-targeted therapy, mTOR-directed therapy, and checkpoint inhibitor-based therapy in non-clear cell kidney cancer. And if we lean on real-world data sets like this, I mean, gosh, IO-based therapy looks really good, right? I mean, I'm not showing you numbers at risk here and so forth, but at first glance, the IO-based cohort here seems to be doing really well relative to the others. But I think the devil with real-world outcomes is always in the details, and there's a very eloquent discussion that accompanies this manuscript. And what they point out there, and I think this is really important, is that patients included in the ICI group were more likely to be enrolled in clinical trials. The proportion enrolled in clinical trials in the ICI group was 55.3%. 55.3%. If you look at the TKI group, it was actually just 11% of patients. So again, I think that this does not necessarily reflect real world if half the patients in the checkpoint inhibitor treated experience were actually receiving therapy in the context of clinical trials. This is likely very carefully selected data here. And what Miguel will show you later in the day is that what we are doing in clinical practice, this rise in use of TKI-IO and what I consider to be perhaps better real-world data, doesn't necessarily translate into clinical outcomes. So stay tuned for that presentation later today. So in summary, I think we really do need rigorous standards for the treatment of non-clear cell kidney cancer. I think that what Tom and Tony and others have shown is that phase 3 trials can be done in non-clear cell kidney cancer, and in my opinion, really should be done to define standards. I think we really do need to challenge ourselves. And I'd suggest that industry collaborations are really essential to increase the speed and efficiency of drug development in this disease space. Thank you very much.
Amazing. Thank you so much, Monty. That was a wonderful talk.
The theme of the session is bridging biology to breakthroughs. So I really think we're, in my opinion, many of these latest trials that you pointed out, Monty, have really at their core really drug approval strategies rather than really making a breakthrough based on biology. So I hope, I mean, it behooves us now in academia and with the FDA that we really get back to not having another drug that's a me-too drug or another combination that really doesn't add to what we have already, but rather than really focus on, and I think I loved the presentation of Vivek about you know, how we can move the needle forward with tumor tissue agnostic to try and really to identify targets and really move the field forward with real breakthroughs.
Thank you so much. I don't know if anybody from the panel has to comment on that. And I think it was really exciting to hear Lisa and Srini's talk on just where are those new targets and where the field's going.
Maybe just a quick counterpoint. I agree with some of what you said, at least Nazar. But I will say that fundamental principles in oncology, right? We really want to understand what the true contribution of components is. We really want to understand what we're really sort of observing in phase 2 studies, whether or not that translates into the context of a randomized design. I would suggest that everything that we're seeing in the context of TKI-IO trials in non-clear cell kidney cancer is certainly encouraging. But I would, actually applaud our industry colleagues for addressing what I think is a necessary question, which is, is there really benefit there over TKI alone? So trials like Stellar 304 will address that. Molecular selection, right? We think there's benefit there. Is there really? I think Sumanta will address that. So these are necessary questions, I think, that industry is helping us tackle.
From a patient advocacy organization, we see it as a lot coming down to funding, especially nowadays. So it's really important to have strategic partners. And also, I would say, the CDMRP, just to give another plug, has been an incredible partner to us. Has funded a lot of incredible research into VHL and to CCRCC. And if anybody's interested in learning more about how you can get funding for your research and how we could, VHL Alliance could help advocate for you in your research through CDMRP, please come up and talk to me.
And I think that terminology, which was perhaps used as a variant, came from or emanated from urothelial carcinomas, which do have possibly some variants, but then there's a common cell of origin in urothelial carcinomas, and there are common underlying genomic aberrations, and over there perhaps makes more sense. But in kidney cancer, in non-clear cell RCC, the genomic drivers are different, the CNVs are different, the cell of origin is different. So actually, we clearly know non-clear cells are not coming from a clear cell. So I think we might, this is a very important meeting, so I think we might want to stay away from the variant histology term. Then I think again, from a pathology perspective, I think it's great to see there are so many pathologists in this meeting. My request to everyone who's leading clinical trials, which there are so many over here, is to make certain that there's an experienced geopathologist on all these clinical trials, which I think can be really helpful from a clinical trial perspective. And then in referral centers, like in our hospital, we are seeing a MITF RCC case or a NF2 mutated or Merlin deficient case almost every month. So what I'm trying to say is, like, while these cases are uncommon, they are not uncommon. Till a referral center is involved, till people can do the correct interrogation to reach to the final diagnosis for these cases. Like, our approach is, if we do not stop interrogating a tumor till we have exhausted all resources, including genomic resources. And the last suggestion or recommendation is to, which has been already alluded to as well, is to perhaps to make certain to include a genomic arm on all of these trials. While histology counts, there are tumors which go beyond histology, and I think there will be more definitively diagnosed by genomics. And I think that has been alluded to in the several talks as well. Thank you.
Excellent. Another comment here?
Oh, Kim. Kim.
Yeah, that's actually a great segue into the, I wanted to make a comment and then ask a question for Dr. Weinstock. So first is, this is a great session, and I think thinking broadly about how we handle rare tumors is, in the clinical trial setting is something we really need to talk about and be thinking about how we do it differently. The comment about that we need to move from genomic definition to AI to help us is absolutely true. So the thing that keeps me up at night is that all cancers are becoming rare cancers, right? As we're sub-segmenting and we're defining them better, all of this is getting harder for everyone. So kidney cancer actually has an opportunity to be a real leader because we're only one of the only tumor groups that has had this as a part of our makeup for a really long time. Okay, so number one, so that I think we could really lead in this space. Number two is that I just want to share an anecdote about how we move, as Vivek was just saying, to like truly all hands on deck is an anecdote from pancreatic cancer. So at The Ohio State University at the James, we have an investigator who's taking pancreatic cancer subset that has FGFR mutations. There are oral FGFR inhibitors out there, and has this really innovative clinical trial that identifies those patients through their genetic sequencing that's done anywhere in the country, right? So identifies them, they don't come to Ohio State. They are managed remotely. You know, telehealth visits, connection with their local doctor, drug is shipped to their home because FedEx goes everywhere, and managed in a completely distributed clinical trial design. So that's a way that you can take something that, you know, in a pancreatic cancer setting, even at a big place like Ohio State, might have only seen 5 patients a year, would have taken, as you said, 10 years to accrue this study. We really think we can accrue this study in a year. So, you know, we can really think outside the box.
We have about 5 more minutes.
Could I?
Please.
Hi, I'm the patient advocate for translocation RCC. I have a question for Joshua and Dr. Pell. So how can the patient advocacy involve in terms of getting the industry support for the clinical trial, including the preclinical testing, the design of clinical trial, and the last one, very challenging, how to include the younger children, younger patients in the clinical trial. Thank you.
I think it really, from the advocacy organization standpoint, it has to do with this trust that the patient community has in us and communicating available trials that are recruiting to them. Most people in the community, and I imagine most other rare disease communities, don't necessarily know what's out there. Like somebody mentioned earlier, in the belzutifan phase 2 study, there was only 61 patients. So in a rare disease community, you have so few patients to begin with. So a lot of it from the patient advocacy organization's point of view is communication and marketing opportunities to patients and families.
Yeah, not too much to add to that, but I would say you're the perfect example of, you know, how advocates can participate in really enhancing clinical research, in the sense that I think that you were part of the inspiration to sort of incorporate translocation RCC in Stellar 304, right? And, you know, I think conversely, you also played a huge role in helping us sort of distribute knowledge about that trial to the broader community. So I think if we could follow the paradigm that you've established more often with Joey's Wings, I mean, I think that that would go a long ways.
Thank you.
I did want to actually just maybe, you know, ask a question of Kim, if you don't mind. So, you know, this innovative trial design with the FGFR inhibitor in pancreatic cancer, using the telehealth model, what is the level of robustness of the clinical data we're getting from those sites submitting data via telehealth, in the sense that I think about trials like SWOG Pragmatica, you know, led by one of my dear friends, Karen Reckamp, and this was a trial in the non-small cell lung cancer space looking at pembrolizumab versus docetaxel, you know, in the refractory setting, and you know, the trial ultimately was negative, had the primary endpoint of overall survival, and at the end of the day, you know, in that study, They only captured grade 3, 4 toxicities. There was no capture of PFS, no capture of response rate, et cetera. So we really don't have any, granular information, no biomarker data, et cetera. So I think there was a big trade-off, in a trial design like that.
Yeah, I think there are some trade-offs. I think we have to just choose the right trials for these kinds of approaches. I don't know the nitty-gritty in this trial, but I know that they are measuring response based on scans that are submitted from the outside, and they're using laboratories from the outside. I think it also does speak to, we really do need to look at what laboratories we're requesting. Pragmatic trials have shown that sometimes we don't need all the laboratories that we build in. There's sometimes more bells and whistles than is necessary, but it definitely will lend itself to more pragmatic type questions, but drug repurposing type of approaches and data that may have some imperfections, run on different quality of CT scans and those kinds of things, but that's the real world. And those are the patients that we're trying to reach ultimately. So I do think it has to be designed so that you get an answer. But there are answers out there that we can get.
Great. I think we have...
Kim, just a quick question.
Sorry, Hannah,
go ahead, Hannah.
I just want to mention that it's actually a guidance mentioned for decentralized trial design for clinical trials that would be decentralized elements that we could for the FDA website.
Thank you. That trial that you were mentioning, is it only being conducted in the state of Ohio? Because it gets into the question of licensure across state lines.
You have to have licensure in every state to be able to do that. There's that added. But the compacts cover almost every state in the country now. So it's not as onerous as it used to be to have licensure. I would love to advocate for national licensure.
Yeah.
So.
Yeah, totally.
Yeah.
Last question.
I'll speak on behalf of many early career investigators in the audience and ask each of our panelists, are there any specific advice or opportunities that you have for early career investigators to partner with your specific specialty? So maybe if we start with Wanling, what do you like to see from an early career investigator who wants to partner with a statistician in designing a trial for rare kidney cancers?
So I think the collaboration with a statistician to design a trial. Also, for any trial design, the design is driven by research question. So I think really we need to find a balance between scientific rigor and the practical constraints. So we need to design a reasonable trial in the real world.
Yeah, Monty, of course,
So
absolutely.
So this is actually something that came up at our last SWOG meeting, right? Somebody posed the question of how do I get involved? What trial should I pick within this plethora of studies I could get involved with as a junior investigator, right? So in SWOG, for instance, our high enrolling studies involve frontline kidney cancer, right? Adjuvant therapy for kidney cancer, non-muscle invasive bladder cancer, and frankly, in those spaces, it's very, very hard to compete against the top enrollers, right? For instance, for adjuvant studies, right, it's not unusual to have, a competitor at a different institution who enrolls 50, 60 patients on a clinical trial, right? On the other hand, if you choose to really sort of build a career in a platform, it has to stem from genuine interest, right? This can't just involve, careerism alone. But if you actually, sort of plant a flag in a rare histology, it's a place where you, as a young investigator, can really make a dent. If you have an enrollment of 5 or 6 patients onto a non-clear cell kidney cancer trial, that's a huge dent. I'll use PAPMed as an example. Right? So PAPMed had 152 patients enrolled, right? If you look at the distribution of sites that enrolled patients, about half, right, about 75 patients in that trial came from sites that enrolled just 1 patient alone, right? If you look at, sort of the top enrolling sites, we had 20 from City of Hope. It was my trial, so I enrolled a lot, right? Naomi Haas had 8 patients. And then we actually had a cluster of sites that enrolled 5 patients apiece, right? A lot of Ben Mon at Utah, Tian Zhang at UT Southwestern, and a handful of others, and they all got their name on the publication, right? So, I actually think that planting a flag in these rare tumors is a great thing for a young investigator to really separate themselves from the pack.
Thank you, Monty.
I would just also quickly add that the VHL Alliance offers an early career investigator fellowship. If anybody's interested, please let me know and please apply. We would love to have you join our ranks.
Very
All
good
right.
point. All right, well, I think we're going to
Yeah.
close it out.